Good evening, everyone. Thank you very much. At the outset, I thank organizing committee for inviting me and giving me opportunity to, share my work that I have been doing for the last more than 1 decade. So, going to today, many talk has been already given, but especially I want to concentrate on the Northeast chili, which is already 4 Northeast chili already have been got GI tag. First, coming with the bird eye chili, capsicum proteins, then came Naga king chili, then came to that, Dali Korsani, Sikkim chili round, then recently 2001 that Sirara Kong that used for coloring. Next, please. So, this is GI tag already have been given. Next, please. Next. So this has been already done. Just I want to show that instead of common chili, that capsicum annam, I have been mostly working on Nordic chili. This is bhujolokia or ghost chili. Next, please. So why ghost chili is important? Because 2007 it became the world famous because of highest pungency containing chili, natural local chili in the world. So by the time, many, scientists from abroad came, and before that time we didn't have GI tag or other things. They have taken away all, and n number of commercial products have been already produced by them, Western scientists. And if you go to Western country, you will get chili sauce, many other products utilizing that ghost chili. Next, please. So if you look at ghost chili bhujolokia, you will find in Google like this, like everywhere ghost is given. Actually, the Assamese word bod means it's big size. Earlier people used to say it's big size, but it became bhoot to now become ghost, all famous nowadays. So if you look at here, you will find like this, and if I cross 5 to 6 chili here, you'll be sneezing, coughing, all these things. My students have a tough time to extract the RNA or some other DNA from the fruits. Thank you. Next, please. So there are many uses already, previous speakers have already told, out of which I want to highlight specifically, since it's Northeast pride, bhujolokia, and I belongs to the Assam. That's why traditional use, and since, when we were very young, my parents used to give, with the curry, if we have fever or something like cold, and after drinking, taking that curry, you will be sweating and all the, fever and everything gone. Next, please. So, it not only known for the pungency, it has created many world records, including the 2009 when the famous UK, this one, cook came, Gordon Ramsay. This lady Ananita, she rubbed 51 ghost chili on her eyes and 21 sea opals, became world record. Recently in 2020, Jack Ma, he had in 2 minutes 240 grams of the chili, and the right side, bottom left, he tried to create, world record. He had, ghost chili, but unfortunately he had a hole in the esophagus. That's why he had to admit in the hospital. Next, please. So in 2009, since 2009, it became the world famous, Defense and DRDO, Defense Research and Development Organization. They have adopted this to develop chili grenades. And next, please. Next. So this is a successful example. In 2015, it came in the paper when one Pakistani terrorist entered Kashmir, and he was firing with AK-47, hiding in a cave. And in Indian Army tried to fire back, but they couldn't do. What they did is that with ghost chili, chili grenade, they fired back. After 10-15 minutes, they didn't get. That is what the paper reported. Then when the Indian Army went there, he was almost crying without looking at anything. He was not able to say anything, so he was caught in 2015. Next, please. So if you look at commercial food market, you will get in UK and US like 100 ml of that chili sauce, $11.7 to $12 or pounds or something like. Still you will get. Next, please. Many uses it has. You have commercial food market. In Japan in 2009 and 10, because that time dollar price was Indian currency was less, so $1,000 per dry chili of bhujolokia sold in 1 kilogram. Next, please. So again, many silly shows in the world. So chocolate in any product that people are using ghost chili. Next, please. So vodka also these days it's very available. People are using. Next, please. So just many n number of commercial product. Next, please. So I'm rushing out because of no time. Next, please. So this one Naga practitioner developed Ayurvedic formulation based on the ghost chili, and it was huge success, and she could earn lots of money. Next. So this is how cocoa melt. This girl has mixed bhut with cocoa and strike commercially she could exploit. Next. And you know that nowadays it's being used for the cancer also, anti-cancer, and mostly northeastern India is known for highest occurrence of cancer. And one of my colleagues tried to see whether consumption of very highest pungency or spicy food is correlated with cancer, development tumor. But our observation in mice found that even though we use this carcinogen along with the Bujuluki treatment, the tumor size reduces in vivo in mice. Next, please. So my first thing was that it has been created lots of world record. I wanted to see whether all the pungency level that has been already reported are still maintaining in capsaicinoid already. But out of this many, these are the different components of capsicum. There are around 14 alkaloids has been reported, but out the 7 are found to be very important. And what we could try here is we can detect only capsaicin, dihydrocapsaicin, not dihydrocapsaicin, because we have standard for the next, please. So first we try to different capsicum species, ghost chili, which is capsicum chinese, and capsicum protein, bird chili small one, and capsicum annam, and bell pepper capsicum this one. What we found from our GCMS analysis is that even though it has been reported as the highest in pungency containing chili, we found from 3 lakhs to 10 lakhs, whereas capsicum proteins 1 lakh to almost 5 lakhs, and capsicum annam it varies up to 2 lakh 50,000. Next, please. So now that time it came in 2005 that PAN1 is responsible for controlling the pungency. Next, please. So if you look at here, pungency biosynthesis, biosynthesis part 2 is that Korean group in 2014 reported, and 2009 Mazurek they have reported that this is the biosynthetic part of capsaicin, and those red colored genes are found to be very, very important as per their report. Next, please. So in 2014, when Korean group sequenced the capsicum annam common chili genome, they've reported that some of the genes are not expressed in non-pungent, like capsicum synthase or 83, then AMT in the later stages, and BK genes. Next, please. Next, please. So since 2012, there was no genomic information, no genome sequence information. Why we have taken this? Because it has been reported that all the pungency biosynthesis genes highly expressed at the gene at the 20 to 25 stages and divide. When you see that capsicum chinense and capsicum proteins having very high pungency content, those red color genes are we have selected and try to see whether those genes are involved in the capsaicinoid biosynthesis genes, and we found that some of the genes are in fact they are expressing very high. Next. So we tried to see in the first report in 2016 we published. So all the genes we have taken in 2 capsicum chinense at that 2 in 3-4 stages, leaf, early stage, breaker stage and mature stages. And first 2 genotype it is capsicum chinense, Bhujologia ghost chili. Second is middle is capsicum proteins, and the last 2 is capsicum annam with low pungency. When you observe this, we found that almost all the genes are found to be very highly expressed in ghost chili genome fruits. That's why our potential, our thinking is that because of this high expression of those genes, we are, the ghost chili fruit is giving collectively high, alkaloids. Next, please. So now, when you look at by the time that 2016, many genome sequences are available: capsicum annam, capsicum chinense, capsicum pagatum. We try to see whether because Stewart et al. They reported only one gene is responsible for capsaicin production high, but when you look at the genome, we found that all genes has many multiple copies. It's very complicated. Now we have to see which gene is really expressing high in case of capsicum chinense, which is very high pungent, compared to capsicum, annam low pungent. So we try to see in all the paralogous genes. We found, next please. So we map in the chromosome, capsicum chinense and capsicum annam. First, we try to look at the already reported QTL region, whether they are matching or not, because we didn't have population that time. Next, please. So next, this is capsicum annam and capsicum chinense. Next, please. So now, very, very less differences are there between different paralogs. Means 83.1, 83.2, 83, there are very, very less differences. We cannot identify them. So we have identified some of the exonic region to see whether, we can differentiate PAN1 and PAN2, PAN3 or something like. So we have designed and QRT PCR, quantitative real-time PCR, whether those genes are staggeredly increasing or they are, expressing very high in different fruit stages in different, ghost chili and capsicum annam. Next, please. So what we found here, if you look at this is just for example, and BCAT, it was reported that it is not expressed in low pungent by genome sequencing, RNA, this one, expression analysis, and AMT, and then 83. The 3 gene only I have taken for example. Then it found that in ghost chili, if you look at here different fruit development stages, we find a very highly expressed. Not only that, only one PAN1 is responsible from this result. Not only one PAN1 is responsible for making the ghost chili more pungent. We presume that this expression of these many genes, many paralogs in ghost chili is important for producing more alkaloids. Next, please. So by the time recently allele PAN1 and PAN3 gene has been found to be expressed, but there are 6 PAN genes which are found to be present in any of the capsicum genome. Now we are trying to analyze how they are expressing. For example, 2 genes PAN1 we have already shown from our earlier slide. So, and not only the structural genes that are involved in biosynthetic pathway, there are genes, transcription factors in which helps in the expression or suppression of the genes, and this one of them is the MIP genes. MIP genes as transcription factors, and in 2019 that they have reported that MIP31 is responsible for expressing of more pungency containing genes. That's what we try to see that from our transcriptome data, because we have medium, low, and high pungency transcriptome data from different fruit developmental stages were there. Next, please. So we found that some MIP genes are very highly expressed in the high pungent ghost chili compared to capsicum annam. Again, we try to see whether those transcription factor genes are highly correlating to high expression of the capsaicinoid biosynthesis gene or not. We have selected only those MIP genes we have shown very high expression along with the high expression of capsaicinoid biosynthesis gene. Next. Next, please. This is what we found co-expression. Next, please. So we try to see from transcriptome data we got very high expression. Now we try to see quantitative real-time PCR. We found that MIB100, MIB16 and MIB31, which already reported by Zhu et al. Are expressing very high along with the capsaicinoid biosynthesis gene. Next please. So we have tried in different germplasm also. We found a similar result. Next please. So again, we found in the QTL region, the black QTL, that black bar indicates the QTL region reported by some other studies, but it's not necessarily that what we get from the QTL region will be directly influencing our trait, because it depends on the genotype to genotype. Next please. So now one way is that we try to see the protein level also, whether the RNA we observe expression in the ghost cili in different fruit stages are also expressing at a protein level. We found that MIB100, for example, here, we found that it's highly expressing. If you look at the, this is transcriptome data, it's found to be correlating with expression in ghost cili. Next, please. So we tried to, functionally characterize those MIB genes, 3 MIB genes. We found that through agrobacterium mediated transformation, we found that expression of high, next. So this is the expression of MIB100, MIB16, MIB106, which is transformed from Burjolokia to low pungent capsicum annum. If you look at that overexpression line, few lines are found to be highly expressed in the genes. Genes are highly expressed, but some lines, capsaicinoid biosynthesis gene have not been found to be highly expressed. Next, please. So when you look at the pungency level of the transform plant, we found that increment in the pungency level compared to control plants. So we have selected, we have found that some of the MIB genes which found to be highly expressed at fruit level may be influencing the pungency development through the capsaicinoid biosynthesis gene. Next, please. So now, finally, molecular marker development, unless it is useful for, to farmers or breeders, it's not useful for when you spend lots of money. So we try to develop and through resequencing sequence of 14 of germplasm, through DD-rate sequencing or whole genome resequencing, we have developed markers, out of which I am presenting only the gene-specific markers today. Next, please. So, next, please. So this is the, 96 germplasm, capsicum germplasm we took, different pungency content. Next, please. Capsicum annum, capsicum proteins, capsicum, annum, and cyanins. So this capsicum cyanins having high pungency, annum low pungency. The last is capsicum proteins. We wanted to see whether the molecular markers are linked or not, whether we can get all the cyanins will have same molecular type or different molecular type compared to the capsicum annum or capsicum protein. Next, please. So we developed like this intel marker based on whole genome sequencing and gene sequence we get from the capsicum cyanins, capsicum annum, capsicum baccatum. We developed in intel markers. Next, please. Next. So what do we see from here? Adjustment. So what do we see from here? The first few, gel pictures are from the capsicum annum, and second and third are from capsicum cyanins and capsicum proteins. These are capsaicinoid biosynthesis gene-specific indel markers, because we have identified SNP marker, we have identified SSR marker flanking the genes or upstream 5 kb or 5 kb downstream of the genome of the capsicum where the genes are lying. So what we found that whatever high expression found in the capsicum cyanins and capsicum proteins, high and medium pungency, it is found the same because there is no difference between annum, cyanins and proteins. Only difference is between the annum and cyanins. So it shows that, and Bosland also, they have reported that cyanins and proteins are very closely related. That's why maybe this is the reason. So we develop indel marker, gene-based indel marker. Next, please. So we develop transcription marker also based on the gene itself, which are showing very high expression as well as which are found to be related to QTL already reported. But still we have to test. This is only the species-specific marker. Still we have to see whether all the high pungency are having the same marker for annum and cyanins and proteins, because we have developed only specific markers. By this, we can predict that these genes from the capsicum cyanins ghost-cilic can be, interrogated to the capsicum annum low pungency. Next, please. So this is again we try to the in the population this is Dudu is capsicum annam and Lota is the capsicum chinense, ghost chili, high pungent more than 7 lakhs. This is around 30,000. So we genotype all the markers that we have developed, Indian markers. We found when we associate and statistical analysis, we found that those markers are associated with the pungency development in the pungency contained in the progeny. Next. So we have developed this marker also. We have tried in different markers. Next, please. So finally, we have many markers developed, but I have not shown here due to limitation of time. Genome-wide marker, SNP marker, as well as SS marker, and mostly we targeted that this indels because very easy to genotype in agar gel. Sometimes SSR also become very tedious when the size is very less. So I thank Department of Biotechnology for Government of India, SERB University Commission for funding the research. As well as these are the students who did all the work. That Ilyas Ahmed, he did all the marker development of capsaicinoid biosynthesis genes. Sarfraz, he did all the capsaicinoid biosynthesis first paper. Susil, he did the transcriptome, Abdul transcriptome marker development. John Momo did that protein analysis of placenta and pericarp and try to correlate whether sequence variation, RNA expression, as well as that protein expression, whether it is correlating or not. Kusbu Islam did that MEP transcription factor. Next, please. So these are some related publication with respect to this. Thank you so much. I hope I have not taken much time.
Lot of work. Okay. Thank you very much. I think we are at the end. I know we have very few people left in the audience. Anybody? I don't think so. Dr. Arjun. Oh, Madhvi. Dr. Madhvi.
Yeah, hello. Where is he, sir?
One question you have.
Yes, ma'am.
There's a question for you, Nirala.
Yes.
Oh, very nice presentation. Congratulations. I wanted to ask you, within the species, if they're low, medium, and high pungency,
Yes.
will it be possible to...
Madam, that we have not
You
looked
did between the
species, but
within annum.
within annum actually we got all the same annum. The marker profile is same, all annum.
You're not able to...
We have not able to, but
Is it possible to do some...
it's very difficult, madam. That's why we were looking for the upstream and downstream of this one.
That's what we also didn't find.
Yeah.
I thought you are basic science. No?
Yeah. But number of gene combination that what you have seen, if you make a cross between Chinese and Annam, maybe number of gene that we have shown, transcription factor as well as the gene indel marker, you can find maybe that we have not done till now.
Within the species, it will be more useful to the chili breeders.
Yeah, within the species, it's very difficult. Now we are looking for that because they have. We are using only the one capsicum annum reference genome,
Okay.
the Korean group. So our Indian genotype is not sequenced fully till now.
Okay.
So we have not tried it. Also, I am just concentrating on that ghost chili and then bird chili.
Thank you.
That will look for that. Thank you.
Thank you. I think you can take it forward outside for more discussion.